Vanakkam. Welcome to this course on classics in neuroscience. We have been looking at neural circuits. In the past bunch of videos, we looked at the spinal circuits, we looked at the retina, and we looked at some invertebrate models and central pattern generators. In this series of videos, we look at cortical columns and cortical processing. In the last video, we looked at the work of Laurent Dino. In today's video, we will look at the work of Vernon Mountcastle. In the 1950s, there was an explosion of interest in using microelectrode recordings from cells, not just in the spinal cord, but also from the cortex, from other regions of the brain. One of the earliest pioneers in this line of work was Herbert Jasper, was a colleague of Wilder Penfield in Montreal, and the other pioneer in this line was Denis Albe-Fassard from France. Although these works were carried out in the early 1950s, the key breakthrough, the major breakthrough came from the work of Vernon Mountcastle, who went on to discover the cortical column. So, Vernon Mountcastle, to give a little background about Vernon Mountcastle, he was born in 1918 and went on to medical school. After service in the Second World War, Vernon Mountcastle went on to work with Bard, who was an authority in the field of emotions and neural representation of emotions. So, he worked on neurophysiology in Johns Hopkins, and he diverged to continue a different line of work, that is on neural responses to tactile stimuli, touch receptors. That was his first line of work. So, Mountcastle recalls the dramatic day on which he discovered, or he figured that there was this columnar organization. Right. He says that, I am going to read from the text. So, he was studying the results of tests on the brains of cats, recording the character of each cell from successive penetration layers. I was writing them down vertically on a yellow strip of paper, yellow piece of paper. Suddenly, the vertical note taking helped him see the stunning pattern in the brain. Skin cells lay atop skin cells, joint cells lay atop joint cells, and so on, extending columns from the brain surface all the way down through the 6 layers of cortex. He says, that was my aha experience. So, that is the time when he thought, oh, so this is what is going on. So, critically in all his experiments, the microelectrodes were inserted radially. So, let us say this is the surface, the insertion was always, let us say this is where he is inserting, the insertion was always perpendicular to the tangent at the surface, that is radially like that, or rather like that. But if you were inserting like this, for example, obliquely, the results would be different. So, in all his experiments, Mountcastle inserted radially, and he found that cells that were responding to a certain type of stimuli were present in the same column. For example, cells that respond to light touch were present in the same column. In a different column, cells that responded to deep touch were present, and cells that responded to joint movement, for example, were present in a different column, but they were all present in the same column, for example. So, there was penetration of these electrodes, in the radial direction into the cortex. So, cells that fire impulses for same type of skin stimulus were present in the same column. But suppose you are inserting obliquely like this, then you will cross through different columns and you will find cells that respond to different types of stimuli as you are inserting. So, this means that there is an organization that is along a column or along a radial direction, something to keep in mind. Because of course, this is not entirely new, because we have seen in the previous video that the work done by Dino, where he also suggested something similar in his contribution to the physiology book. So, this is not entirely new, but this was among the first evidence to suggest this. So, in his 1957 paper, Vernon Mountcastle outlined his observations from the somatic or somatosensory cortex. He found that the elementary unit within the somatosensory cortex was a vertical group of cells, and this vertical group of cells essentially extended through multiple layers in the cortex. So, it is not just present in one layer, it just goes deeper, 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 deeper. So, it turns out that cells within a column within this vertical group show similar discharge properties. What are the similar discharge properties? They respond or they fire or they discharge at more or less the same latency, and they respond to approximately similar or same stimulus, and they respond to the same or nearly the same peripheral receptive field. Of course, it is likely that there was an effect of anesthesia, because these experiments were on cats, on anesthetized cats. The more complex responses were perhaps reduced, were perhaps inhibited by the effect of anesthesia is one possibility. Of course, this was not deep anesthesia, just light anesthesia, but still it could cause this kind of an effect, something that we can expect. Of course, it does not mean that these cells are activated monosynaptically, although this is one of the possibilities that was outlined by Dino. It is not necessary for these to be activated monosynaptically. There can be interneurons that can happen. The point is that cells within this column respond to similar stimuli, have similar receptive fields and respond at similar latencies, but cells between columns respond to different types of stimuli with different latencies and may have different receptive fields. So, that means the organization is columnar within the cortex. So, this is the takeaway message. So, within a vertical column, synaptic connections of cells of the ascending axons, these make up a large percentage of the cells in higher mammals, those that are studied by Mountcastle. This indicates that there is going to be, it is possible that there will be multiple impingements of these. So, it was also shown that synaptic connections within the vertical column of the ascending axons, these are very powerful, and these essentially make up a large percentage of the neurons in the cortex, in higher mammals especially. So, there are these cells that make synaptic connections, powerful synaptic connections along the ascending axon within the vertical column. These make up a relatively large proportion of the cells. It turns out that they make synapses with cells in all the layers, across the layers, or with many levels of depth they may make. So, there might be multiple impingements of this information that is going to various layers, something to keep in mind. Of course, this idea is not new. I have already mentioned this. This idea is not new. This was somewhat similar to the work done by Dino, or the suggestion made by Dino. Meanwhile, Roger Sperry's experiment involving vertical subfield dicing of visual cortex showed that very fine visual pattern discrimination was still retained, suggesting that organization is happening along vertical columns. Similarly, vertical dicing of sensory and motor cortices almost did not affect normal function. Once again, confirming the notion of existence of this columnar organization within the cortex. Later, Tom Powell from Oxford joined Vernon Mountcastle in Johns Hopkins, where he devised a method for identifying the tract or the path taken by the electrode as you penetrate, both in radial and oblique penetrations, and the cells that are innervated are measured by this electrode. All these studies essentially confirmed the columnar organization of the cortex. Importantly, it is crucial to note that the anatomy of the cortex is tied to the physiology, or there is a clear structure function relationship here at play here. There was no, by this time after the work of Vernon Mountcastle, there was absolutely no doubt or no questions about the suggestion that there was this organization, this columnar organization of the cortex. This was not being challenged by that time because the evidence presented by Mountcastle and his colleagues was so compelling. With this, we come to the end of this video. In this video, we looked at the contribution of Vernon Mountcastle, especially towards identifying the columnar organization and providing compelling evidence or non-controversial, incontrovertible evidence suggesting the presence of this columnar organization within the cortex. With this, we come to the end of this video. Thank you for your attention.